Let's talk about MHC and antigen presentation. Antigen acquisition and transport. Antigen presenting cells. Structure and function of MHC molecules. Antigen presentation to T cells. Firstly, acquisition of antigen. Foreign antigen will enter the body through the skin, respiratory tract, or the gastrointestinal tract. Cells will pick up the antigen and transport it to the peripheral lymphoid tissues where adaptive immune responses are initiated. Now, adaptive responses. Antigen has to be presented lymphocytes in organized lymphoid tissue by antigen-presenting cells, APC. APCs include macrophage, dendritic cell, very efficient, Langerhans cell. Immature DC cannot activate T cells, must encounter antigen first, digest antigen into small peptide fragments. Dendritic cell maturation. Immature DC will become a mature DC with increased MHC2 and other receptors on its surface, responsive to cytokines and chemokines, involved in chemotaxis. Chemokines are grouped into 4 main subfamilies: CXC, CC, CX3C, and XC. All of these proteins exert their biological effects by interacting with G-protein coupled receptors, chemokine receptors, that are selectively found on the surfaces of their target cells. The major histocompatibility complex, MHC, or HLA in humans. The major histocompatibility complex, MHC, are a set of cell surface proteins essential for the acquired immune system to recognize foreign molecules in vertebrates, which in turn determines histocompatibility. Main role is to bind to antigens derived from pathogens and display them on the cell surface for recognition by the appropriate T cells. The MHC determines compatibility of donors for organ transplants, as well as one's susceptibility to an autoimmune disease. 3 types of MHC molecules: MHC1 and MHC2 and MHC3. MHC class 1 genes: HLA-A, HLA-B, HLA-C. MHC class 2 genes in humans: HLA-DP, HLA-DQ, HLA-DAR. MHC class 3 encodes TNF and complement factors, C2, C4, and factor B. The cleft size is slightly different in type 1 and type 2 SOCs. Type 1 is 8 to 10 amino acids long. Type 2 is 12 to over 18 amino acids long. Major determinants as to whether an organ transplant are accepted or rejected. Organ rejection is normal, if undesirable, as the immune response is directed towards non-self MHC molecules, MHC or organ donor. Role of MHC. A T cell only recognizes antigen when it is presented by an APC that has a particular MHC molecule, MHC restriction. Applies to CD8, T cells recognizing MHC1 and CD4 recognizing MHC2. Some PARAC describes MHC molecules as looking like a hot dog bun. In the grove of the bun lies the peptide fragment, like the hot dog. The T cell receptor recognizes both the bun and the hot dog. Each MHC and CD4 and CD8 receptors. CD4 is found on T helper cells, and CD8 is found on T cytotoxic cells. It is the relationship between MHC and CD4 or 8 which defines the type of immunological reaction which will take place. Antigen processing for presentation by the MHC receptors. 2 major pathways by which antigens are processed and presented to T cells. Exogenous and endogenous pathways refers to the source of the antigen. Exogenous antigen is taken up from outside the cell and presented to MHC2, example is bacteria. Endogenous antigen is made up inside the cell and presented to MHC1, example is virus. Naive versus effector T cells. Some difficulty used to be experienced in interpreting experimental results until it was realized that naive and effector T cells differed markedly. Naive cells are those which are meeting antigen for the first time. Effector cells are those which have gone through antigen stimulation and have become activated, proliferate, and are ready to kill target cells.